So here we go. Hello, everyone, and welcome to phyloseminar.org. The next three talks will be in honor of the late Carl Woese, who I'm sure, as everyone here knows, revolutionized our understanding of the microbial world via molecular phylogenetics. There may be no better person in the world to introduce his research accomplishments to us than Norm Pace, who will be speaking to us today. Following Dr. Pace, Ed DeLong will speak on May 15th concerning Woese's contributions to microbial ecology, and Phil Hugenholtz will speak in June. Dr. Pace had published 267 papers between 1960 and 2010, and he continues to publish at a prodigious rate. His work originally concerned the molecular biology of ribosomal RNA. This led naturally to performing sequencing on these molecules. Comparing these sequences led to a significant change in our understanding of the tree of life. He's also been a leader in bringing this new understanding to the classroom. Dr. Pace has received a great amount of recognition for his work, including becoming a member of the National Academy in 1991, as well as a MacArthur Award. We do encourage questions during the talk. If you have a question, please go ahead and type your question into the meeting message box, and I will read it aloud. All right, Norm, take it away.
Oh, thank you. Well, welcome, ladies and gentlemen. I don't know who's out there or where, but welcome. So this is going to be a talk about two things. One is the beginnings of metagenomics, but I really want it to be a paean to Carl Woese. Carl Woese put in place a new way to think about biological diversity, and no one else was doing this at the time, and it was quite a remarkable understanding. It also revolutionized our views of microbial ecology. So here's the outline I'll use then. You all know what Carl Woese did. He did quite a few things, actually, but in particular, he put in place the scientific tree of life. All previous views of life's diversity and early evolution were basically guesswork. And Woese's tree was the first scientific tree, the three domains tree of life. And because of the impact in microbiology and many other things, it's really created a new biology for the 21st century. So I'll talk a little bit about what Carl put in place and how that gave rise to what we would call modern microbial ecology and or metagenomics. And then I'll talk about how the applications of metagenomics by many people have really expanded pretty dramatically our view of microbial diversity. I'll talk a little bit about, at least my view of the current state of the tree of life and uncertainties that are intrinsic, and of course spark a lot of controversy. And I'll finish up with a public service message from Carl, directly from Carl approximately. Microbial ecology. Microbial ecology is running the biosphere, and we know very little about it. And one reason for that is that microbiologists long had to culture organisms in order to even detect them. And it turns out you can't culture very much of what's out there, and so most of life's diversity evaded us until what we would now call metagenomics came along. If you go down to the banks of Boulder Creek out behind our building, or to a drainage ditch outside your current town and pick up a little bit of the green scum that inevitably occurs and put it under a microscope and blow it up 1,000-fold or so, it will look something like that, or I should say a few hundred-fold. And when you look at this massive, bewildering complexity, you don't recognize much, perhaps a diatom, maybe a premnesiophyte, and a few other things. But in general, it isn't until you go to much higher magnifications and begin to look at all the dots and squiggles and so forth in the background, you realize that it's the little dots and squiggles that are most of the diversity that you view. Well, people have recognized for a long time the complexity of the microbial world, and people have always tried to puzzle out what this meant. Of course, if you're going to do that, you need some sort of reference framework that's meaningful. And of course, the reference framework that people have been groping toward since early in the 19th century, certainly even before that, was what we would now call phylogeny. It's about relating organisms by virtue of some attribute. And like biologists, again, have been talking about phylogeny for a very long time. Of course, Darwin's famous quote is is that our classifications would come to be genealogies, which they are now doing happily. And again, people have been thinking about this for a long time. For example, here's a tree from Ernst Haeckel in 1866. And Haeckel had his monitor down here at the bottom, and somehow they turned into animals and plants and protists, microbial eukaryotes. This was his worldview, and it's fine, and it was with us for the next 100 years. And about 100 years down the line, Haeckel's four kingdoms became with the contributions of an ecologist by the name of Whitaker, who believed that fungi were really important, and indeed they are. And he built this five kingdoms tree with his monitor down at the bottom, which somehow turned into microbial eukaryotes, which then somehow turned into plants and fungi and animals. And a contemporary theme, which gave even a larger glance at this, is what was put in place in the early 1960s by Stanier and colleagues, the concept of two large kingdoms of life, prokaryotes, which then somehow gave rise to eukaryotes, just like Whitaker would have said, or Haeckel before him. The problem is that these were subjective views of it all. Didn't really have at that stage of the game any way of having an objective view. But then, with the development of rudimentary sequencing techniques, it was possible to grope toward an objective description of life's diversity. The concept of making phylogenetic trees relating organisms by sequences really came out in the mid to late 1960s, and Woese began to work toward developing phylogeny based on that. Now, you all know what molecular phylogeny is, I'm sure. You wouldn't be tuned into this, but just in case, let me say just a word about it, because my molecular phylogeny, at least in principle, is simple. You get the comparable sequences from different organisms, and then you very, very carefully line them up so that homologous nucleotide or amino acid residues are juxtaposed. Then you simply count the number of differences between pairs of sequences, and that's some measure of the evolutionary distance that separates the two pairs of organisms, the two organisms in any particular pair. And then it's a matter of calculating some sort of a map that would relate all of the pairwise sequences to give a larger relatedness network, and we would call this a phylogenetic tree. Now, you can do molecular phylogeny with any molecule that you want, but if you want to understand the molecular phylogeny of all of life, there aren't many molecules that you can use. And the one which has really emerged as the gold standard and introduced by Woese and developed by Woese and many other people since are the small subunit ribosomal RNA gene sequences, 16S ribosomal RNA, 18S in some eukaryotes. Woese began to study small subunit ribosomal RNAs at a time when we couldn't sequence the entire molecules. Actually, he first began with 5S ribosomal RNA, which you can sequence at that time, although it took about a year for 120 nucleotide sequence to determine that sequence. But Woese soon decided that there just simply wasn't enough information in 5S RNA to relate all of biology, and he turned to the small subunit ribosomal RNA. And that molecule is typically around 1,500 nucleotides in chain length, and at that time, it was impossible to sequence something of that length. Woese, on the other hand, recognized that he could use primitive sequencing technology developed by in the lab of Fred Sanger at the MRC in the UK. And this was a method whereby one would take P32 label RNA, 16S ribosomal RNA, for example, digest it completely with ribonuclease T1, which would result in a collection of oligonucleotides containing A, C, U, and a 3' terminal G. And those could be separated in an electrophoretic pattern, of two-dimensional electrophoresis, separating out all of these oligonucleotides resulting from RNase T1 digestion. Now, one could then go in and extract in these individual spots and subject them to subsequent nuclease digestions and work out the sequences of those. Very laborious job. Woese spent much of the period between about 1970 and 1985 staring at these electropherographs and working out the particular sequences. Now again, Woese couldn't sequence the entire molecule, but he could determine the sequences of the short oligonucleotides. And so he could begin to compile collections of oligonucleotides for many, many, many organisms, collections of the oligonucleotide sequences. And he argued that if he saw in two different ribosomal RNAs an oligonucleotide of six nucleotides or larger in length, then the random probability of the occurrence of those in a molecule the size of 16 sRNA had a low probability of occurrence, and so he could therefore take as homologs of common ancestry oligonucleotides of six residues or longer. And he began to compile these catalogs, and by 1977, then he began to see what was emerging. And what was emerging, of course, was the three-domain pattern of life. Woese's first paper on this, 1977 PNAS paper, included a single table, and this is the table, relating these so-called association coefficients, SAB values, these collection comparisons of these oligonucleotides. And upon doing this, I put the boxes around here. Woese didn't have boxes around them. I think had he put the boxes around it, it would have been a bit more understandable. But if you look in the boxes, you can see that these individual groups of organisms, ribosomal RNA oligonucleotide catalogs, were more like any within the group than any that are outside the group. And this was the first evidence for three domains. He called them three primary kingdoms at that stage of it all. But then by 1990, many, many more complete sequences had come in, and there was considerable correlative information consistent with the notion of a three-domain tree. An origin, what lines spun out, giving rise to bacteria, another line spun out, giving rise to archaea and eukarya. This early life is not organisms of the kinds that we understand today. Just to reiterate a few points that extract from this three domains view of life. One, of course, is there are three main relatedness groups, domains, Woese called them, eukarya, bacteria, archaea. The origin, you can't get it from ribosomal RNA, but the origin of this entire matrix is deep on the bacterial line of descent. And that means that eukaryotes and archaea are related to the exclusion of bacteria. And there's a lot of correlative information about that as well. For example, eukaryotes and archaea have many common elements in their transcription machinery, replicated DNA replicated machinery, and so forth that would distinguish them from bacteria, not to mention other cellular biochemistry. The ribosomal RNA tree showed that the chloroplasts and mitochondria are of bacterial origin. No one would criticize that today, I think. But the eukaryote nuclear line, originally proposed as derived from prokaryotes, it's not. The eukaryote nuclear line is primordial. It's as old as the archaeal line of descent. And this means that this popular prokaryote-eukaryote model for biological organization turned out to be wrong. But for the purposes of this talk and the development of metagenomics, the important point became that the sequences are identifiers of organisms. That is to say, you don't need to culture to identify. All you need are gene sequences, ribosomal RNA sequences in particular. And that gave rise then to the notion of metagenomics. Metagenomics, of course, as you know, they use the DNA sequence information to explore the naturally occurring world. And it's a simple process in principle overall. And there are two general questions you might pose. One question you might pose is what kinds of organisms are out in the ecosystem? So you take a natural sample, you make yourself some bulk DNA and RNA, you use in some way, you make a ribosomal RNA gene library, for example, using PCR, you do a bunch of sequencing, you reflect on the sequence databases, and this gives you a collection of microbial identifications, and so you can now begin to construct the community structure. You want to know, of course, what the community is doing. That's a second question. What kinds of genes are present and active? And you then make yourself a closed metagenome library of every piece of DNA in there that you can amplify or clone. You do a whole lot of sequencing. You again reflect on the sequence databases, and you can now begin to build up the community genotype and construct ultimately community structure and function. This is much more than a game in simply collecting gene sequences. You can do something with the sequences. You can make arrays and do qPCR and fish and track organisms into culture if you want to do something to do that. So now you can go into this complex microbial community that you could only view before, never understand, and can begin to do these things and now learn about who's out there and what they're doing and so forth. Now, this, all of this, of course, was developed subsequent to the Woese development. But in the Woese development, the first rude pictures of the three domains emerged. The only thing that we had with regard to thinking about metagenomics was a natural sample. What now? Well, my colleagues and I at that stage of the game in the early 1980s then began to undertake developing methods for studying the natural microbial world using sequences as opposed to cultured organisms. We could sequence 5S ribosomal RNA, although not yet 16S ribosomal RNA, but we couldn't get 16S ribosomal RNA out of the environment. And so we first went for 5S ribosomal RNA identifications. And we looked at two areas first. One thing we questioned was the nature of hydrothermal vent-associated symbionts, only discovered a few years earlier. These are organisms like Riftia pachyptera, the giant tube worm, for example, that hangs out around hydrothermal vents, or the giant vent clam Calyptrophila magnifica. They were known at that time to have bacterial symbionts that allowed them to metabolize sulfite to make a living, but no one had been able to culture them and find out what kinds of organisms they are. And that, in principle, was a straightforward approach for us. We could go get ourselves a worm and isolate 5S ribosomal RNA from the trophosome, which contains the bacterial components of Riftia pachyptera, the tube worm. We could then label that with label the native RNAs with P32, and then isolate by high resolution gels to separate the quotes eukaryotes slightly larger from the quotes prokaryotes slightly smaller. We could then extract the RNA and then do the kinds of sequencing that you would do at that time, namely end-labeled molecules being digested in limited ways with various nucleases, and again using the RNase T1 fingerprints and other kinds of fingerprints to verify the sequences. And the folks involved in this at this stage were Dave Stahl, Gary Olson, Dave Lane, names that are, I suspect, very familiar to anyone who's looked into metagenomics. Well, this was an exciting thing to develop, but the answer turned out to be boring because the Riftia pachyptera symbiont turned out to be a close relative of a gamma group proteobacterium, a close relative of Phyllomicrospira, and not too far distantly related to our friend Escherichia coli, probably the best known organism. So we weren't finding anything new in this particular setting, but the other setting that we undertook was to start studying high temperature organisms. Same game, go to an environment, this is Octopus Spring in Yellowstone National Park, isolate, in this case, a mixture of 5S ribosomal RNAs, which we could then sequence and determine relationships to other organisms. And I'll mention in passing, Carl Woese was with us on our first expeditions to Yellowstone collecting high temperature organisms. He was very taken with the project of using sequences to study the natural microbial world. Now, with the study of the Octopus Spring, we knew we had it because the sequences that we gathered there, archaeal and bacterial, was different from any other bacterial or archaeal sequences than any were to one another. So this was true novelty. We knew we were going to find a lot of stuff. Well, there was a real hooker, though, because for having the resolution of reasonable phylogenetic resolution, we had to have longer sequences. That meant like small subunit ribosomal RNA gene sequences. And the problem was there wasn't any database, with the exception of Carl Woese's oligonucleotide catalogs. And we could not get that kind of information out of an environment. We needed to pile up gene sequences in some way. Well, and we decided that, well, we already knew from Woese's catalogs that there were some sequences in ribosomal RNAs that seemed to be highly conserved, and with Harry Noller's development and Jürgen Brosius' development of the E. coli 16S ribosomal RNA sequence, we could then lay the Woese oligonucleotide catalogs into that and identify sequences that were particularly highly conserved. And the game at that stage was to accumulate ribosomal RNA sequences, and we wanted to do so by isolating bulk ribosomal RNA from any cell of choice, and then using universally conserved priming sites for reverse transcriptase sequencing. And the folks involved with that, beyond Gary and Dave Stahl, again, Dave Lane, who passed away last year, by the way. Anybody who's used the Lane mask for working with 16S ribosomal RNAs, those Dave Lane effects. Mitch Sogan, And my ex, Bernadette Pace, who's actually a high-flying trapeze artist as well. At any rate, it became possible then to accumulate ribosomal RNA sequences very rapidly by sequencing directly the ribosomal RNAs. So now we were moving along, and we could go directly then to sequencing ribosomal RNA genes out of the environment by cloning genes, first done with the marine picoplankton community, in this case with Ed DeLong, Tom Schmidt. Well, we could now go into the environment and determine sequences, and we wanted to close the loop. What do these things look like, these sequences we are determining? And that was a game of developing phylogenetic stains, fluorescently labeled oligonucleotides, which were generally applicable to organisms such as bacterial-specific, archaeal-specific, eukaryote-specific, or more specific things. In this case, for example, distinguishing a symbiont called sunkiller from its closest relative. And the folks involved here were Ed DeLong, here seen at a Halloween party, Gene Wickham now at Purdue University, and Steve Giovannoni wasn't on this paper, but he was doing related things at the time. So we can now go back into the environment and see what these things look like. And so everything is in place now to begin to explore the microbial makeup of the environment into the natural microbial world. And there are a couple of strategies. First, we use take a sample, make DNA, make RNA, a ribosomal RNA the ribosomal RNA gene PCR library, or directly clone the ribosomal RNA genes. The library would then be cloned. The sequence is done using Sanger technology. And now, of course, when we take a sample, prep some DNA, and do next generation sequencing, getting very large numbers of sequences, a problem in its own right, as I'll comment on. So we and many other people did this for many years in many places. We've studied hot springs in Yellowstone and elsewhere around the world, submarine hydrothermal vents, unusual environments, this being in a block of crystalline salt, endolithic communities, a lot of places in the world, mines and caves, pathogenic microbiology, the open ocean is mentioned, the New York City subway, and even your shower curtain biofilm. Yes, indeed, that soap scum on your shower curtain is actually a biofilm. And of course, the sequences available expanded very rapidly for doing phylogenetic assessments. Just to pick this one, for example, here are bacterial ribosomal RNA gene accumulations for environmental sequences and for cultured sequences. At this stage of the game, environmental sequences far outweigh culture-based sequences in both bacteria and archaea, and certainly in microbial eukaryotes as well. And this, of course, resulted in a pretty massive expansion of known diversity. So when Carl Woese published a rather famous paper in Microbiological Reviews in 1987, and at that stage of the game, he could articulate 12 main phyla of bacteria, all had cultured representation. By 1997, we were up to about 3 times that number. Now only about 20 or so had cultured representation, the rest uncultured, and now we're up to about 100 or so bacterial phyla. Only about 30 have any cultured representation at all. The rest are uncultured, represented only by sequences. And most of the sequences are represented by only 7 of these bacterial phyla. These are the ones that contain pathogens. Now, when you think of bacterial diversity, it's a very much more complex place than names of microorganisms lead you to believe, because any representative of any particular bacterial species, it doesn't matter what, will be carting around a collection of genes all of which won't be present in other representatives of that same species. And this is now being referred to as pangenome. Genes are moving around fairly freely in the environment, and any particular collection of organisms of the same species will have, each representative will have only a fraction of those genes. For example, if you look at 20 E. coli subspecies, they will share only about 30% of their genetic complement. 30% of the individual genetic complement will be seen elsewhere in biology. So the availability of all these sequences refines our view of phylogeny, and so, but still at the large level, at the broadest level of phylogeny, there's still a lot of controversy. Here's what the ribosomal RNA picture looks like from my view. Bacteria, as already mentioned, there's about 100 main phyla, something like that. In my opinion, there is no resolution at the base of the bacterial tree. It's in essence a polytomic. Archaeas, the phylogeny of Archaeas, still a work in progress. The early ideas would have two groups of Archaea, the Crenarchaeota and Euryarchaeota, but as sequences have accumulated, it's gotten rather more complex at the base of Archaea, and then that too, I suspect, will turn into a polytomy. Eukaryote topology is controversial. Many would argue anybody who does ribosomal RNA will see this kind of picture, a deep radiation of some sort, one line of which radiated, one line of which radiated, giving rise to the crown group of animals, plants, fungi, alveolus, spermatophiles, and so forth. Now, this is by no means a universal picture, particularly for folks who like to worry about eukaryote phylogeny, which is a very controversial place, but there's also no real consensus on what it is. Here's one of the popular trees shown for eukaryote phylogeny, and I see that there's much speculation in this. For example, what's this stuff branching off two different directions? What is this thing? In any case, this is clearly a controversial area. I don't want to bang on any drums with it, but we'll see much more of this in the future, I'm sure. Now, there are a lot of problems with resolving deep branching topology. Again, it's a controversial area, and much of the controversy is methodological, and I just want to touch on a couple of things. The big problems in resolving are, one, representation of sequences that are being considered, representative of a large group, and the second thing is intrinsic uncertainty in any phylogenetic tree. It gets more uncertain the deeper you go into a tree. Representation is a big problem because of all the sequences that we have represent only a few of the microorganisms that are out there. So, for example, if you look at the bacteria, there are about 100 of the bacterial phyla, but most of the sequence representation is found in only the most abundant half dozen or so. And once you get beyond that, there's very little sequence representation. Unless you have appropriate sequence representation, you cannot generate a good tree topology. In the eukaryotes, it's even worse. Of the many eukaryotic sequences that are out there, they've been derived from very few of the clays, mostly fungi, a lot of animals, a lot of plants, a lot of alveolus, some spermatophiles, which are then there. That is to say that microbial eukaryote phylogeny is approximately not resolvable at this time with any meaning whatsoever. Now, one important point, of course, is increasing that resolution, but right now, operationally, we've got a problem just because of the nature of sequencing. Next generation sequences are short, and accuracy is problematic for the regions of the ribosomal RNA that are being read. And so if you look at the large scale of ribosomal RNA accumulation in the databases, yeah, it was really going up for a long time, but now it's pretty much shut off because the sequence is too short to even go into the databases. You may get a taxon call, but not a very good resolution at all. Moreover, the pipelines, the sequence processing pipelines, in dealing with what some would call pyrobabble, they toss out the new stuff that don't fit the training set, but that means you throw out the new stuff. But I hope it's getting better. We're doing this as well, and you do what you can do. The other thing I draw attention to in any phylogenetic analysis is intrinsic uncertainty in any particular branch points in trees. And the uncertainty is greater the deeper you go into a tree. And this is an issue of unseen change. Namely, if I compare two sequences, I might look at C and A and a G juxtaposed. There might have been ancestral A, but I don't know that that was a single step from an ancestral A to the observed G. It might have gone from an A to a T to a C and then to a G. So there were unseen changes that you never saw. Now, there are ways of estimating these unseen changes, a Poisson estimation, but nonetheless, they are estimations. And so if you consider any collection of paired sequences, your observed change accumulates in some way. The observed change is such and such difference, but the actual change, because of this nonlinear dependence upon unknown materials, unknown change, the deeper you go into a tree, by the time you get to the domain level of variation, 30% of your signal is guesswork. At the phylum level in the bacteria, say 15% of your estimate of branching order is guesswork. That's a real problem. And again, with sequence length and so forth, that will improve over the years. If you make progress, more long sequences of phylogenetically diverse creatures, obviously genomics will have had high impact on this. Better treating algorithms are still needed for exploring deep phylogeny. Some of the methods that are out there are clearly wrong in the conclusions that they drive and result in controversy. And a really interesting question, of course, is how to go deeper than the last universal common ancestor. And indeed, that was one of Carl Woese's white whales, I would call it, one of the many things that he thought about over the years. Now, this is the final component of this talk, and I'm going to go to a subject which was very dear to Carl's heart, as he pointed out in the 1977 paper that the previously believed notions, the common wisdom of prokaryotes and eukaryotes or 5 kingdoms, he pointed out that those were just wrong. And so my public service message from Carl Woese is this: there ain't no such thing as prokaryotes. It doesn't exist. It's a bad idea. It's a bad model. And this is about more than names. This speaks to the very essence of biology as a science. I think that there are 2 fundamental issues, and only 2 fundamental issues in any scientific discipline. One of these is order. What is the organization and the relationships of the elements that you would study? So, for example, the chemist would study the periodic table to understand relationships between the atoms. And we biologists would study phylogenetic relationships to understand relationships among the many diverse organisms. The other thing that any science worries about is change. How do those things that you would study evolve? How do they change? So the astronomers, for example, would study the Hertzsprung-Russell star series to understand how their favorite star fits into the evolution of stars. Biochemists would study biochemical mechanisms to understand how CO2 turns into us. And molecular phylogeneticists, of course, would study the molecular tree of life to understand the course of evolution. So there are 2 conceptual models of biological organization and evolution that have emerged. There's the classic textbook tale. Even now, there was an origin which somehow gave rise to a radiation of prokaryotes, which then somehow turned into a radiation of eukaryotes. And that's in contrast to the observed 3 domains, which we've already talked about, namely an origin giving rise to a line which ultimately differentiated to diversify as bacteria, and a second line which then diverged into archaea and eukaryotes. Those are diametrically opposed in terms of the way that we need to understand evolution. So a very big lesson from the big tree, Carl Woese's big tree, is that this prokaryote-eukaryote model that dominates our textbooks, dominates the discourse in microbiology, at least for a long time. It did so, still does for some folks. It's wrong. It's an incorrect model, and incorrect models never take you to truth, of course. Now, if I'm going to tell you that something is bogus, I also have to tell you where it came from. I've actually already told you where it came from. Right there is where it came from. It came from Haeckel's monera from the 19th century, converted into prokaryotes 100 years down the pike. Prokaryote means non-eukaryote, nothing more. And of course, not being something is not a scientifically valid concept or name. Now, let me bang on this one with a bit of a hammer by testing directly the formal model that was introduced by Roger Stanier and colleagues in the early 1960s. He fobs it off to Edouard Chapon in the 1937s, but it turns out, as Jan Sapp has pointed out, that this publication doesn't exist. The 3 precepts as articulated by Stanier for the prokaryote-eukaryote model are: 1, all eukaryotes are of a kind, specifically related to one another, represented within that wedge. All prokaryotes are of a kind, related to one another, to the exclusion of eukaryotes. And finally, that prokaryotes gave rise to these more advanced eukaryotes. We can now test that model because we now have data, as Carl Woese provided to us. Here's the Woese 3-domain tree. All eukaryotes are of a kind, specifically related to one another. That's true. All eukaryotes would fit into that wedge. All prokaryotes are of a kind, related to one another, to the exclusion of eukaryotes. That's false. There are 2 of them. They're prokaryotes, one of which is more closely related to eukaryotes than it is to the other one. That's false. That's not with phylogeny. And the final idea that prokaryotes gave rise to eukaryotes is also false. The eukaryote nuclear line of descent, in contrast to the organellar line of descent, the nuclear line of descent is as old as archaea, primordial. That's a very different way to think about the evolution of eukaryotes, and it is the proper way. Why am I doing this to you? What does it matter if we call them prokaryotes? Who cares what the name is? Well, I care because that name prokaryote is scientifically unjustified. It was invented and accepted to fill a gap in knowledge. We didn't know what that little stuff was, and so that term prokaryote was invented to accommodate that ignorance. And indeed, that name has false implication in deep evolutionary matters. This false understanding of prokaryote quenches legitimate inquiry. You would be hard-pressed to find an NIH panel that would fund a grant on Sulfolobus acidocaldarius, the archaeon, even though the biochemistry you might want to study might be very much more similar to our own than to that of the model organism E. coli. But here's the one that really bums me out badly. Prokaryote-eukaryote model of evolution teaches our students false concepts at the very most fundamental levels of biological organization and evolution. Very bad to impart bad information at fundamental levels. Well, what else to call them? I'm often asked. Well, if you mean the little stuff out there, to try microbes or microbial. There are a lot of little eukaryotes out there too that are perfectly interesting, and interestingly sort of fell through the cracks of prokaryote and eukaryote. Eukaryotes are so often being taken as plants and animals. The microbial world of eukaryotes is a very interesting place. Vanished to the students. And of course, if you're talking functional or evolutionary issues, you definitely need to be more precise than prokaryote because that is a failed model. Remember, to students, concept is built by language. Therefore, the language builds the understanding. Therefore, it's got to go. Bad understanding, bad model. And with that, I'll offer final paean to Carl Woese. So thank you, ladies and gentlemen.